right, thanks everybody for joining us here for the weekly IDMP series, which is focused on COVID-19. This morning, one of our associate members, Ben Gerwitz, who has been studying viruses all his life, is actually going to host our special speaker this morning. So I'm going to turn it over to Ben.
Okay, thanks, Deb. So it's my special pleasure to introduce our speaker today, Dr. Mark Denison. Mark is the Chief of Pediatric Infectious Disease and the Craig Weaver Chaired Professor at Vanderbilt Medical School. And whereas many of us have become coronavirus researchers for about 3 months, Mark has spent his entire life in science studying coronaviruses, I think beginning this as a postdoc at the University of Iowa with Stanley Perlman. And since then, he's developed a world-class program at Vanderbilt focusing on coronavirus replication, pathogenesis, and countermeasures. And interestingly, his first manuscript on a novel antiviral for coronaviruses was published already about 25 years ago, targeting the viral protease, demonstrating truly remarkable foresight. The seminar today is quite timely because Mark stuck with that pursuit. And among his many achievements, he is the person who brought Remdesivir to the coronavirus field. He had a major role in developing it and even had a role in designing the trial. And as you've probably heard through the news, it's now the only drug to have FDA emergency use authorization for COVID-19. Tony Fauci recently declared that it's the new standard of care. And I saw on the news today that it was just approved for use in Japan. So Mark, I know this is an incredibly busy time for you and just want to really thank you for joining us here today.
Thanks, Ben, and Marissa and Deb and everybody for inviting me. I appreciate it very much. It's a real honor to be here. And I guess I live a little bit in hope right now. If I think a Remdesivir story is anything, we don't have the data out yet. As a scientist, I'm very cautious. As a physician, I am hopeful, and as a human being, I am emotional, I guess, about it in terms of the possibility and opportunity to at least contribute. We know that that antivirals are not there's no such all of us who do science and medicine and training understand the word cure is not a word that we should apply to these things. Our expectation is to be able to potentially intervene, prevent severe disease, help those who are in severe disease survive and recover, and to also, but at least at a minimum from the science part, to be able to recognize that there are potential mechanisms that we can do to really combat this in association, obviously, with other causes, things like vaccines, monoclonal antibodies, and obviously essential need for public health measures. So with that, I'll start. I hope you all can see my slide. Am I shared? Make sure I'm shared here. Apologies.
It's working.
Okay. All right. Please let me know, Ben or others, if you can't see what I'm sharing right now. So my talk today, coronavirus replication, development of Remdesivir and other nucleoside analogs. I realized even that was like 3 talks: replication, Remdesivir, and others. So we'll have to settle for something, and I expect everyone to go away a little disappointed, but I hope at least a little educated, understanding some of the vulnerabilities of these viruses. But before I start, I want to make sure I recognize the team that's been working on this for really a long time. Maria Agostini, who's a graduate student who has been working on this and is leaving. Today is her last day in the lab, going to work for Vera Biotechnologies in California. Andrea Prousers, who is running our program, and then really everybody else, Chatelu, who's been working with me for 30 years. Brett Case, who is now a postdoc at WashU, who did the very first experiment with a Remdesivir precursor, and Erica Andres, who is just starting her residency at Penn, who, as a technician, did remarkable work that really set the standard for how we were going to work with this drug. And then many others in the lab who have participated as well. And I also want to thank our other collaborators. Obviously, UNC, I've been collaborating with Ralph Barrick and his team for the last 20 years, and really with this one particularly, as well as the team at Gilead Scientists, who are the discoverers and developed and patented and own this. And if you hear anything about them that suggests a non-humanitarian purpose to this, I would argue vehemently against that, because they maintained this program and worked with us to supply the drug and to help develop the mouse models that were necessary for testing it in mice over a 7-year period, when there was nothing except a few cases of MERS here and there and no SARS and very little interest in this. So in fact, if anything, I would represent them as extremely humanitarian oriented and powerfully supportive of this process, as well as our funding at University of Alabama, Birmingham through a U19 that Rich Whitley PIs, and then our other grants as well. So I wanted to be sure I recognize that at the beginning. So this is just, I have comments from our U19 application, our project from 2018, and I could show the same one from 2013, actually, from the first iteration, that the epidemics of SARS and MERS in humans have confirmed the capacity of coronaviruses to emerge from animal reservoirs and cause severe human disease. SARS-like bat coronaviruses are currently circulating in nature that replicate in human cells, cause disease in mice, and are resistant to existing therapeutic antibodies and vaccines. And with increasing overlap of human and wild animal ecologies, the potential for severe zoonotic COVID emergence is high. And of course, we were just parroting what many people, like the people at EcoHealth and others, have shown from their exceptionally important work with bats, and looking at bat ecology and bat evolution, as we all know, is critically important work for us to be able to predict. But we and others, Ralph Barrett probably did some of the first work back in before 2000, showing that you could adapt a mouse virus to hamster cells. And once you adapted it to hamster cells, it became a generalist that was capable of infecting a broad range of cells. And so the coronaviruses had that potential even before the SARS epidemic. So each time this happens, we're shocked. I'm utterly shocked, but I'm not surprised. And I think that the potential in coronaviruses is high for this to happen again. If we think about this as a sort of a 1,000-year flood, that means there's a 1 in 1,000 chance it'll happen again next year. The odds may be even higher. So I think we need to be thinking not just about what we're dealing with right now, as terrible as that is, but also how we look at this to prepare for things that could happen in the future. And that future is very much with us when we think about the emergence of coronaviruses with pandemic potential. Shown on this graph, or this line bar, is sort of obviously the estimated archaeophylogenies, I guess, thinking about where these viruses came from, and just showing the endemic human coronaviruses that cause respiratory infections, cold, and sometimes pneumonia and more severe disease, but that are endemic. The most important point I'd say from this is even old coronaviruses are new. That is, even if you think about 700 or 800 years for a brand new human disease, that's recent. And that there's ones that are as recent as the 1950s. Likely all of these had a bat origin initially, and then went through other animals. OC43 is most closely related to a bovine coronavirus, and to murine coronaviruses. The other ones are H229E is an alpha coronavirus, probably mostly closely related to a bat virus. So, and then when we look at this accelerating emergence with SARS in 2002 to 2004, MERS in 2012 to 20, and then obviously SARS-CoV-2 now, we can see these increasing pace. I think that this represents probably not anything fundamentally different about what's happening with these bats in nature. It reflects our intrusion into ecological niches, wildlife marketing, increased demographics. I can't directly blame climate change, but I think anything that is going to affect the ecologies and the interactions of these animals and humans is going to be a factor. I think there's no doubt now that this is a high potential, particularly for these group 2B beta coronaviruses in the SARS-like group, to do this and have devastating disease. I just want to show something here. I'd love to spend a whole hour talking about this because I think it's critical, but since we can't, I want to point out a couple things. One is this is sort of the overall this is an old phylogeny, and I just put boxes on it, so there's not any indication that these are bootstrapped in any way. But I want to say that if we think about SARS-CoV-2 and SARS, and then if we look, BT means bat, and bat viruses, there's well over 200 bat coronaviruses has been identified. Really every genera and species of bats that's been looked at are shedding a coronavirus or 2 or 3 asymptomatically. So there's this vast reservoir. The question of exchange between species, it may not occur very readily. Bats are independent species. There's over 1,300 bat species, 22% of mammals are bats. They are critical and they circulate either widely, some migrate, others are very local and do not circulate widely, but they interact. And what we can see is the human coronaviruses really are coming from multiple groups, and they all within each of those have bats within each of the groups, the 2As with human coronaviruses and our model virus, MHV, which I'll talk about a little bit. And then, but this group up here is really the place where we think we're dwelling right now. MERS is from this group here. But you can see that the potential for these is high. The potentially and this has been a little controversial, but exceptionally important. This idea of identification of bat viruses, and this was done by Minicheri and Ralph, when he was in Ralph's lab, looking at these bat SARS-like viruses, which were reconstructed in the lab and then tested for their capacity to infect human cells and animals, and showed that capacity right out of the blocks. So these are viruses that just what Ralph likes to call ready-made pre-pandemic viruses. No modifications, no change. And I think this was the big shot across the bow, which suggested it. Unfortunately, leading to controversies, but unwarranted ones. But nevertheless, really was what was helping us to predict and say that we needed to be ready with antiviral strategies. So what's the need for antiviral strategies? Well, there's a long list. I think we can talk about the advantages and the disadvantages of these, but mostly the broad diversity of bats with demonstrated capability to infect human cells in animal models. The time to develop vaccines differs from trajectory of epidemic. So it would be hopeful that we could find ways to have universal vaccine strategies can be immediately implemented, but they still would be a new virus that would have to be tested in humans. And so the concept of having something off the shelf toward highly conserved functions that's sort of future-proof is critical. What I don't go into here, which I think about a lot, is the idea that, as you all know, people sort of like to think about winners and losers. So you'll see this thing, failed Ebola virus drug used in SARS-CoV-2, which is all kind of nonsense. Right? The drug worked in vitro exceptionally well, and it probably worked in humans, but the nature of the disease, and it wasn't as efficacious as other modalities. So I think we need to eschew this sort of dichotomous thinking, and really move toward a model that's more about understanding when, how, why, and where compounds would be efficacious and work. For example, the idea that we would have compounds that would work like Remdesivir, that's IV, that would work in patients who are ill, progressing from disease. So to prevent severity, prevent progression, and to rescue from severe disease. Right? Potentially, it could be used as an outpatient in an infusion setting if you had someone who was at high risk, right, and didn't want to come in the hospital. But that's just one approach. Ideally, you would have inhaled versions or versions that were PO of drugs that could be used earlier or to prevent disease, or used in a nursing home where people have been exposed and you're trying to prevent progression and severe disease, so in a prophylactic manner. And also, if you find compounds that really were good at decreasing the specific infectivity of viruses, their capability of actually causing infection. So it sort of act as temporary attenuating strategies. So viruses that come out are less capable of causing disease. You might be able to make some at least local or regional interventions in hotspots, or sort of like putting fire extinguisher on a brush fire that's popping up as you're trying to deal with the conflagration that's occurring. So I think our strategies really weren't drug development strategies, and when they started, and so I've been learning drug development, as I call it, a second language late in life. Yeah, I'm immersed, I'm learning it. I'm really excited and passionate about it, but I haven't been doing it my whole life. So there's a lot of things I don't know. And so we've come at it from the virologic and biologic perspective, and I've decided to be intentionally naive about drug development and focus more on the virus biology, because I think that's where the new outcomes will really come from. So our goals for antiviral development, as I said, were slightly different from those of others, and that is one of the first things we wanted. We came at this with the virus biology, so we wanted something that absolutely was broadly active against diverse coronaviruses. So as we begin to look at hits or screening or drugs, if we'd start with mouse hepatitis virus, our model virus in BSL-2. If it was positive, we'd immediately test it in MERS and SARS in vitro. If that was positive, then we'd immediately work with Ralph and collaborators to test it in human airway epithelial cells, primary cells, and then move on to animal testing as quick as we could. And it had to pass all those milestones of efficacy against multiple coronaviruses before we'd be interested, because otherwise it suggested to us a cell specificity or a virus specificity that might limit its ability to be used in the future. High barrier to resistance. So we also, particularly if we're talking about single drugs initially, we wanted limited genetic paths so the virus could not quickly escape. And if the virus escaped, that there was a high fitness cost. That is, that if you got a resistance mutation, that the virus was less fit and less able to survive in absence of the drug. extended therapeutic window. You all are probably know more and read more and have seen more than I have as a pediatrician at Nashville to think about the immunologic complications, the immunopathogenesis of this disease. So one of our goals of our grant was actually extend the therapeutic window. In other words, be able to co-manage direct virus replication and cytopathology, along with immunopathologic consequences and the late implications of disease. So for amelioration, for treatment, for prevention, all these things are critical. So and to say that also, combinations for any future purposes, or even for this one, those should be done. The second you have one drug, you should be looking at it with other drugs. And that's challenging, as I'm discovering, as a NIH-funded lab scientist. That's challenging to get people to cross their boundaries between their companies, between their own motivations, and to get into a different mode of thinking, which we all need to be in during this pandemic. Of first, let's look at the good of humanity and see what we're done. And you can tell I trained as a pediatrician. That's just how I view life. And so I think those things should all be done. And obviously, then I meant to decrease oral transmission of the virus. So, I'm afraid I'm preaching to a choir and people who are more informed about this than me. But I will deign to insult you a little bit, I guess, with simple stuff for a few minutes. I think the approaches we can think about countermeasures in general, direct-acting antivirals, so those that act against the virus target, or theoretically a host target that is antiviral. I'll just call them antiviral strategies for treatment, prophylaxis, and transmission, and we'll get to that with Remdesivir. And monoclonal antibodies. So I love developing these drugs because I love the virus biology and want to try to kill it and target it. But if I think where the promise is, I would hope the promise is in monoclonal antibodies that we could use for temporary or indefinite, I would call it, passive immunity that could be used to block virus entry in the first place, stop its replication, and be able to help those who are vulnerable to have some temporary immunity while we're working for other strategies. And then host-directed therapies to modify or block disease. And finally, what I don't have up here is vaccines, which are obvious and critical if we're trying to develop any kind of durable herd immunity, even locally. I think thinking about 7.8 billion people needing to have herd immunity to this virus before we get this under control is a daunting concept. So that's when I wake up at 5 in the morning thinking about that process and where we currently are in that process. But we won't go there because it's early in the morning. So we'll stay on the positive. And Ben, and those others who are monitoring, if there's questions or chats that come up along the way, I'm happy to address those. So I, of course, start with the virus biology. And my world is, like they say, is I've learned more and more about less and less till I know everything about nothing. But we're really focused on the replication strategy, particularly that in cytoplasmic. So I just want to review this very, very quickly. I know the grad students and all of you are very, very good, but I'll go over it again for those who may not know this. So coronaviruses, as in all RNA viruses, are obligate intracellular pathogens, right? They replicate in the host cell cytoplasm, and they have nothing to do with the nucleus, at least from the perspective of entering into the nucleus for their replication strategies. There's likely nuclear proteins that are associated in the cytoplasm with viral replication complexes. But virus entry for SARS-CoV-2 is mediated by a binding of the spike glycoprotein to the ACE2 receptor and entry. And coronaviruses broadly entry, we don't really know this yet about this virus, enter by two mechanisms often, and they can swap those depending on the circumstances. One is endosomal mediated entry. So they go into an endosome. There may be modifications within the endosome, which allow for the fusion protein of the spike to be engaged, and for fusion, the virus cytoplasmic fusion to occur, virus membrane fusion. The other is a directed through mechanism, binding and fusion at the cell surface. This may be very well dependent on exogenous proteases or endogenous endosomal proteases like TMPRSS2, which may modify or change, as well as acidification of endosomal lysosomal fusion events that allow for the virus to enter. So what happens after that? Well, once that happens, these are positive strand RNA viruses. Their genome is a large, massive mRNA, one of the largest known, certainly the largest virus mRNA. And so it's no DNA in their life cycle at all. They enter into the host cell. And the next step after entry then is going to be translation of the genome. And 2/3 of the coronavirus genome is translated into about a 900,000 Dalton protein that contains 16 protein domains, a string of pearls, 16 proteins, 2 proteases for SARS-CoV-2 within that string of pearls, and then it begins to autoproteolyze itself into mature proteins. Those mature proteins then, from the polyproteins, then modify, actually the next step is actually modification of host membranes into replication factories, where you get assembly of the replication proteins, including the polymerase and the exonuclease that I'll describe. And then you get replication and transcription of a series of RNAs that are used for making structural and non-structural proteins. And then the genome, copying of the genome. Virus proteins are modified on the ER, and assembly occurs in the ER Golgi intermediate compartment with a path involvement of multiple secretory and maturation pathways, glycosylation steps, and as well as then assembly into sort of typically vesicles, secretory vesicles that release large amounts of virus. And viruses aren't, at least with the models and the ones we're aware of, are not immediately profoundly cytopathic or cytolytic, and they tend to release massive amounts of virus without really destroying the cell. So that's why they can really put out lots of virus without other damage. So the step, alas, I'm skipping over, and my apologies, but you're going to get what I know. And that is really this focus on this issue of virus replication. So this is really where, as Ben said, I think I spent my whole life. It does kind of feel that way a little bit sometimes, certainly since 1984, working on this. And the first, I can say I for this one thing because it's what I did, was the first to describe a coronavirus processing product from the replicase genome. And since that time, I've been interested in the viral proteases, the polymerases, and my lab team and our collaborators have really focused on this aspect of it, intracellular replication as a target. So this idea of the processing, protein processing, assembly, RNA synthesis, and this is really where Remdesivir lives. So this is why I could spend time on this. Particularly focusing on the polymerase as really a highly critical protein, one of the most highly conserved. So when people are trying to look at phylogenies and clocks, they'll tend to look at the polymerase for 2 reasons. One is it's highly conserved across the viruses. So if it's across diverse coronaviruses, you may have 50% conservation of a sequence, or but in the polymerase, you have between 80 and 90%, even across the widely diverse ones. And so variations that occur within that region can give you a lot of information because they're rare. They're not common within a group. Okay. And so polymerases are actually structurally conserved. It's just hard for me to keep up, so I don't have the structures that have already been published on the SARS-CoV-2 polymerase, which are elegant and doing. But this one I want to emphasize because Rob Kirkdorfer, who's at Wisconsin right now, published this first one for SARS last fall, and this was really the first cryo-EM, really beautiful structure which showed the complexity of it. And I wish I had time to go into how polymerases work, but I don't today. We'll just talk about it from the perspective of the drugs. And so nucleoside analogs, obviously, and base analogs are efficacious antivirals against multiple RNA and DNA polymerases. These are chemical modifications and imitate normal nucleoside RNA nucleosides, and have been some of the most effective. Obviously, acyclovir is the model by which the altar at which we all bow in terms of the first and sometimes still most effective, remarkably, after all these years. Richard Elliott and others, it's just beautiful work, amazing in helping this model. It's sofosbuvir, which is a Gilead product, and as one of the first and highly efficacious ones to actually, I guess we can call that cure because it's possible to cure hepatitis C. So that in combination with other drugs is remarkable. Ribavirin, which I say everybody likes to try against everything, despite its toxicities and oftentimes evidence that it doesn't work, because it works in vitro against pretty much every virus, except coronaviruses, which I'll show you. And then others like favipiravir, which a lot of stuff is coming out, at least on preprint, saying yes, it works against flu, but no, it doesn't work against coronaviruses. So, what do we do? Well, we had tested. This was sort of like, they say doing the same thing over and over again and expecting a different result is a definition of insanity. Silly statement, but people tend to do the same experiment whenever there's a new coronavirus. They tend to use ribavirin or other drugs. And And we had never seen it work in vitro, and this is sort of our first blush. And so when Clint Smith, I'll tell you where this came from. Clint Smith, assistant professor of biology now, did this work. Basically, the first simple thing he wanted to do as a postdoc was say, let's just look and see how these drugs work. So what you see here is this is log reduction of the virus on the y-axis, reduction in virus titer with increasing dose of drug. These are really high concentrations of drug. And the theoretical, ideally, of course, would be increasing drug, decreasing virus replication, up to 4, 5, 6 logs. Well, I think it's relatively straightforward to see that this doesn't work, no matter what you do. And then using chemotherapy, using 5-fluorouracil, which is a mutagen. So ribavirin probably does chain termination and mutagenesis and multiple other things, but none of those were effective against the virus. 5-fluorouracil is a mutagen, a high-level mutagen, which really knocks down lots of different viruses, and no effect out to 200 micromolar, nothing. Okay, and even with the theoretical. So the question was why? Well, we kind of knew the answer, but I'll act as if we didn't. And so back up just one step, and you've heard this also, that RNA viruses have low fidelity replication that compared with DNA-based organisms. So when you think about DNA-based organisms, you're talking about up to 10 to the minus 10th mutation. So one mutation in 10 to the 10th bases replicated, right? So very high fidelity. And that involves basically the polymerase, which has sort of a base. And this is a misstatement of many people, that RNA virus polymerases are error prone. Well, every polymerase is error prone. It has to be, or it can't succeed. It has to be fast enough, and so it's going to make mistakes. But what do other organisms have? They have proofreading, which is recognition and prevention of incorporation of mismatched nucleotides. Then they have later mismatch and excision repair, which really provides the highest rates of fidelity. Whereas RNA viruses live in this domain without proofreading, and their range, so you can see that their fidelity may be, a millionfold less, and overall. And so this is they generate mutations on a regular basis. But we were always puzzled by the size of the coronavirus genome and wondering how this could be. So here's something from Sasha Gorbalenia, which it's still so pretty that I just use it all the time. And that is that if you look at RNA virus genomes and their genome size up to 20,000 nucleotides, what you see is all these plus-strand RNA virus genomes of the coronaviruses, the togan flaviviruses, they all live in this domain between sort of 5 and 15,000 bases, nucleosides. And then there's over here, there's something. So way out here in the 30s. So what is this? So what's the difference? Well, the difference is you don't see any nidoviruses here. And nidoviruses are the order that coronavirus is in. But if you add those onto the chart, what you find out is there's one family called the arterioviruses that's small. But look at this, there's this sort of like this evolutionary gap. And over 20,000, the only viruses that have been found, at least that infect humans, that are single-stranded plus-strand RNA viruses are in the nidovirus order, including the coronaviruses. And the difference between those, and giving it away, is that they encode a novel enzyme, a 3' to 5' exoribonuclease. Even without knowing the function, that's just a difference that these other viruses don't have, and is thought to have been the reason that they've evolved large genomes that are stable. So invite me back for another talk on that sometime, but we'll talk a little bit about it, which is that the coronaviruses encode this exoribonuclease, and we took this data and decided we wanted to understand it better. So this is the virus genome, the full virus genome, with this replicase polyprotein that I mentioned, this sort of 900,000 Daltons of protein encoded, including multiple replicase proteins, and specifically the RNA-dependent RNA polymerase. And then here's this novel non-structural protein 14 exonuclease domain. And that was recognized by Gorbalenia for having these motifs, so DEED motifs that he thought looked like an exoribonuclease. It's in the DEDD family of exoribonucleases, which are proofreading enzymes in DNA-based organisms and implicated in high replication fidelity. So we wanted to understand what the impact of this was. Could we knock out this enzyme and could the virus survive? Well, multiple people had tried in coronaviruses, but we were able to do it in MHV and in SARS. And basically by making two amino acid substitutions and 4 nucleotides, and then rescuing this virus from an infectious clone. we were able to rescue virus that had a replication defect, about a 1 log replication defect. So it was remarkably robust in its replication. And then what we did is we wanted to know how many mistakes does it make. And just to summarize it, what we discovered with MHV and identically with SARS was that this virus inactivating the exonuclease resulted in a mutator phenotype with 20-fold increase in replication in mutation frequency. Or the flip side is that the wild type coronavirus, and when we measured this, what we found was that this mutation frequency really was closer to other RNA viruses, whereas in fact, the exonuclease was providing about a 20-fold increase in replication fidelity. So this is, to date, still to my knowledge, the first and only known RNA-dependent RNA proofreading enzyme, using RNA as a template. So this is, the textbooks haven't all yet been modified, but this has now been confirmed enough that I think what we can say is coronaviruses have evolved a proofreading system, which is fascinating for many reasons. But when we measure it repeatedly, what we find is this tends to put coronaviruses right in this range, right here. So they don't have mismatch and excision repair, at least not today, but they do certainly are capable of having up to potentially 100-fold increased replication fidelity. So what has that got to do with drug development, and how did that lead us there? Well, it was really this, I'll tell you one anecdote. It was really this story that led us to studying Remdesivir, getting Remdesivir, because I was on sabbatical at Pasteur Institute. I actually decided to go to a coronavirus meeting. I was the only coronavirus poster at a coronavirus meeting, and was showing this work to Craig Cameron, who's chair of micro at UNC now. And he said, gosh, you know, I've been working with these compounds from Gilead, and I might put you in touch with them. Maybe there's something that could work against this activity. And so he did. And when I got back from sabbatical, after about a year of what I call the junior high school dance, who's going to dance with whom, with Gilead, we started working together, and they let us start to look at this. But so the question is what does this have to do with nucleoside analogs? Coronavirus is likely assembled this multi-protein replicase holoenzyme that looks probably more like a DNA Pol III. It looks more like a giant multi-protein DNA polymerase holoenzyme with the polymerase exonuclease, processivity factors, helicase, unwindase. So what's that got to do? Well, remember I showed you the graphs with ribavirin and 5-fluorouracil. So when Clinton finished this experiment, he tested it against our exonuclease minus virus and saw that these were profoundly sensitive to nucleoside analogs. So it wasn't that coronaviruses weren't sensitive to them, it's just that they had a mechanism for preventing it. And so the question was it possible to get something that could block this proofreading activity? So we then did, and got those compounds from Gilead and started our experiments. And so just to let you know, these processes, nothing is magic. Gilead, a little bit, it looks like, gosh, look at this drug, it's right here, and all of a sudden it works. Well, we started the first testing in 2013. And then first testing against MHV. And in green is what we did. Blue is stuff that we did collaboratively with UNC. The yellow is things that were done externally. And as you can see in a brief blush, that this really was actually a 7-year process of very careful development of this compound, and met many milestones along the way with our interactions with Gilead, Ralph's lab at UNC, and then the University of Alabama Birmingham with the support through the U19. So this was the money graph that Brett Case did back in 2013, and I show this only because I accused him of making a mistake and being wrong. Remember, the curve was, this is wild type MHV. The curve, remember, that I showed you for those other drugs with wild type virus was flat, and this would have been the exonuclease minus curve. And this drug showed activity, significant activity. We didn't know what it was. We were blinded to it. It was called GS441524, which is the active compound from Remdesivir, but we had no idea what it was at the time. And we said, this looks like this might work. And so what they did next is they sent us about 60 prodrugs, meaning chemical modifications of the drugs that they had on their shelves that they had been working on with their hepatitis C studies. And what we saw was this exceptionally profound increase in activity with all of these. And one of them particularly emerged, and that was GS5734, now known as Remdesivir. That was significantly more active. It's sort of this is a little bit of a cartoon. But if this is the activity of GS441524, this is the activity of Remdesivir. Okay, so it was between 3 and 100 times more active than the parent drug. I was a little enthusiastic at this point, and this is where Maria Agostini, the grad student, picked up the work. So, for those of you that don't do any of this, I'll keep it simple. But I want to say basically what we were able to see was EC50 is in the very low nanomolar range, which is exceptional and rare, and reduction in virus titer of up to 6 logs at really 0.5 micromolar. So a millionfold reduction in virus titer, and beautiful. I didn't know this for a long time, but this is a beautiful EC50 curve with no inhibition to complete inhibition, and demonstrating really the efficacy of this drug against our model virus. So then we tested it against our exonuclease virus. And what's interesting, exonuclease impaired virus. And what's interesting is, yes, remdesivir is very active, but if you knock out the exonuclease, it's even more active. So you can imagine that I'm excited about the potential biology of this, of mechanisms to increase the activity of these kind of compounds by inactivating the exonuclease. So how does it work? Well, it has an oxymoronic terminology, which is called a non-obligate chain terminator, meaning that it causes the growing RNA chain to terminate, but it doesn't have to. But it does. But it doesn't have to. But it does. Okay, but so that's my understanding of the chemistry. That's how complex my understanding of the chemistry is. Essentially what's happened, and there's a beautiful paper out of JBC by Gordon et al. From Matthias Goeth's lab in Canada, showing that what people had predicted, which is known, is that in fact it incorporates as in Denison, and then something about the modification on the 3' location on here allows actually for the incorporation of 3 more nucleotides, and then it stops, which is fascinating. So that's why it's called non-obligate, because it can bond with additional nucleosides, and then it terminates. And what they showed is that if you it's massively favored from other nucleosides. But if you increase the normal ATP to high enough levels, you can abort this termination and you can get extension. But under normal conditions, this compound really terminates the RNA chain. So very effective, not mutagenic, and able to block RNA synthesis at all stages. So our next question was, does remdesivir select for resistance? Because that was our next question. We wanted to make sure it didn't, and what are the consequences? So what we had good news for Gilead when we called them finally. What we said, we have 2 bits of good news. One is that it's really, really hard to get resistance to this drug. We had to coddle it and treat it really nice and keep giving it a break. And it took us about 23 passages, which actually was about 40 passages because we had to start passages over again multiple times. And what we saw was that we could get a virus that was less sensitive to the drug under those conditions. But the resistance at the EC50 was only about 6-fold. And it was always overcomable with higher concentrations of the drug. And what was really remarkable in this 30,000 base virus after 23 passages with the drug is that there were only 6 mutations, and most of those were irrelevant mutations, and only 2 of them were really relevant, and those were both in the polymerase, in the RNA-dependent RNA polymerase, 2 mutations that we identified. And so when we went back and put these back in the virus, if this is wild-type sensitivity, which is very sensitive, if this is the passage virus in green, when we put the mutations in alone individually, we got partial resistance, and when we put them in together, we got almost the same as the passage virus, and which is some interesting questions we want to address. But the point is, they seem to be responsible. But the question we had was, so what? What does this matter? Well, what this mattered because the viruses seem to replicate fine. So we wanted to do competition to say, so how does it compete with the wild-type virus? So the way we did those experiments is just basically a competition. And in this case, we used a ratio of viruses with the wild-type 1 and the mutant 9. So we gave 9 times as much mutant virus in the cell at the same time with the wild-type virus, okay, with the resistant. And so we put them in together. And after 1 passage, it looked like what we put in, more of the mutant, resistant mutant than the wild-type. But as we passaged those viruses multiple times, what we saw was really the wild-type virus dominated. So in more of a sort of a relevant approach, what you see is that the virus ultimately, even though it replicated fine, it couldn't compete with the wild-type virus. And then when Tim Sheehan took this in SARS and put this in the SARS background and put it in their mouse model, and using high doses or high inoculums of the virus, what we saw was that this virus, in blue, was less able to cause disease, and the animals recovered better. So it showed a significant attenuation of virulence also. So we kept getting good news. I kept saying, this drug development thing is really easy. Like, this drug does everything we want it to do. And people said, that doesn't work that way. I go, no, look, I've done 1 drug. It's working every step we take. So not true, but But that was my feeling. N of 1, it worked. So interesting. So a couple summaries. If we start with the parent compound, the active compound, we had significant efficacy. When we took remdesivir, which was the prodrug, which is better metabolized and integrated and more rapidly phosphorylated in the cell, we saw about a 30% increased efficacy. If we took that virus and we put it in our exonuclease virus, we saw another 6-fold increase in efficacy. So we're talking about 180 times here. And then if we passaged it for resistance, what we would see is about a 5-fold decrease efficacy compared to remdesivir. But that virus had impaired fitness and attenuation in vitro. So this is why our enthusiasm was obviously high for this. So I want to just get onto so that's enough of that biology, but just to give you a sense of the animal. So this is why bat virus work is important. Okay. What you can see here is that, remember, we've shown it worked against SARS, we've shown it worked against MERS, we've shown it worked against MHV. But what do we have here? Well, this is a human alpha coronavirus, causes colds. This is a bat MERS-like virus. This is a bat SARS-like virus. And these are those 2 viruses that I called sort of pre-pandemic viruses. And what you can see is the graphs of beautiful work by Tim and Amy Sims and others, showing that this virus, the virus is just as sensitive. These viruses are just as sensitive to remdesivir, same EC50s. So we were most excited about this because of its potential breadth. And this is what made us aware that this might be useful against a new pandemic virus. So just to summarize an exceptional amount of work, why should trials be designed the way they are? And we can hopefully come back to this for a few minutes. Remember, remdesivir is IV administered. So what Tim and Amy and others showed was that if this is given before or early after infection in an animal model, which is highly accelerated in mice for SARS compared to humans, that if you give it really early, you decrease titer dramatically, you decrease weight loss, decrease lung pathology, improve lung function, prevent disease. If you give it later in infection, you still whack the virus, always, okay? But you don't decrease the lung disease or mortality. So this is why, and then there's something out in bioRxiv now from Williamson et al. at Rocky Mountain Labs, showing a decreased virus, fascinating, that IV administration in macaques, which are a replication and kind of a pulmonary disease model, but a pulmonary change pathology model, but not really a full disease model, showed decreased virus replication in the lung, but not the upper respiratory tract with IV administration, and decreased lung virus titer and disease, really suggesting its work and efficacy. So that's all very exciting. So in terms of thinking about this from human disease and human testing, this is why when I was asked my opinion about trials, I said, if you're just going to give it to the very, very, very sickest of people only, then just don't do the tests. Because in human disease, what we need to understand is not if the virus works, but the drug works, but how and where and when it works in human disease. We need to think about a spectrum of disease treatment, from those that are at high risk and might progress, to those that are stable but ill and not getting better, to those that are slow progression and are highly concerning, to those that rapidly progress and are on ventilators, to those that are compellingly failing. And thinking about how we use these drugs will matter. So when I think about remdesivir, I guess the summary of it is that, it potently inhibits the virgin coronaviruses. It has a high barrier to resistance. It's useful in prophylaxis and early treatment in mice and macaques. And then we have now data, some it's out in bioRxiv, that this is highly active and showing the same kind of activity against SARS-CoV-2 in HAE cells, in CALU cells, in mice infected with a chimeric SARS coronavirus with SARS-CoV-2 RDRP, so that we could rapidly do this work while SARS-CoV-2 models are being developed. So I just want to make a few more comments. I want to stop and have some time for questions, and that is that we need to be continually thinking about this. I am I'll step away from my physician scientist and even pediatrician and for a minute and say, this is coming back to my sense of hope about this. So if this was I'm waiting for these results expectantly. I was more optimistic than I thought I would be after even hearing these announcements, which shouldn't be meaningful, because if this was given to all comers, and particularly biased towards severe patients early on, if there was any signal for the efficacy or use in the most severe patients. Then, of course, those numbers about mortality and recovery would be very conservative if you could, in fact, look at patients who are more moderately ill. And I'm hoping that the parsing of the data will allow us to actually see when and where administration of Remdesivir and or other nucleoside analogs or other approaches would be most useful. Along obviously with the goals of multiple DAAs, which we're working on now, and trying to think about combination therapies of direct-acting antivirals, monoclonal antibodies, and immunomodulators, which is work that must move forward. And then oral use or early use, and then targeting different steps. We have to keep focusing on the biology. We have to keep being rigorous in our approaches to testing. We cannot abandon our methodologies that have gotten us here. This was a 7-year process with no expectation that this would ever be used in humans in my lifetime, because that wasn't the goal. The goal was, can we find something that's a common target that will allow us to be prepared and will allow us to have a mechanism for intervening? We have other compounds we've worked on, such as this Emory compound, EIDD 2801, which is orally bioavailable. And then basically I could recapitulate this talk in terms of its ability to work, except that it's a mutagen, not a chain terminator. So I want to stop. I'm sorry, I haven't left this so much time for questions as I wanted, but I wanted to just to let you know that we are really, we think this is really important, and we'll continue to do this work and target new areas. The exonuclease obviously is high impact. Those initial studies that have been talked about in my work on protease inhibitors, starting in 1995 with when we identified the first coronavirus protease, are areas we're going to keep working on as well to try to enhance the possibility to intervene in this particular epidemic as well as the ones to come. So I'll stop with that and be happy to take any questions.
Thanks so much. That was fascinating. It really provides a rich perspective on what's happening right now with Remdesivir and how we got there and where we're going next. So I know there's going to be some more questions from the audience. I thought I would just start out with a few. So one that I was wondering about during your talk is, so how does Remdesivir sneak past the exonuclease activity? Do you have an idea for?
Well, there's some models and some structures that are coming out now, and one suggests that it has this very efficacious binding in the grooves, the RNA binding grooves of the polymerase, and that this mechanism of this sort of, they call it I3, so incorporation and then 3 nucleotides later you terminate, maybe something that hides it enough from the exonuclease. So what's been proposed is that because it's so, I just looked this up at 2 this morning, by the way. What's been proposed is that it's so much more favored over ATP in terms of its incorporation that it gets in, that has a mechanism for getting in the first place. And that because it can extend, that can happen quickly enough the biochemical studies that have come out of Bruno Kennard's lab and others have shown that if you get past 2 nucleotides, the exonuclease doesn't work very well. So it may just have to do with it's like a sledgehammer. It just drives itself in. It's a high-speed bullet that drives into the nucleoside, and then it gets protected and hidden by the addition of additional nucleotides under its normal mechanism, and then termination occurs. That is fascinating. Those are of course models that look at this and are looking at the biochemistry, and so we'll see. But so far the models and the modeling support it. So I think as more cryo-EM and crystal structures come out. The other thing, remember I showed you that model with the the whole thing is this: there may be 8 viral proteins in the replicase holoenzyme. We have to look at it in context of that, and recapitulating that biochemically and by any kind of structural stuff is a Herculean task. But I think with thousands of labs interested, I think maybe we may see progress that we might not have seen in 50 years of this in the next several months.
Mm-hmm. Wow. I was wondering if you think Remdesivir could be converted to an oral prodrug, so it could be given earlier and more easily, and maybe in combination with the other one that you mentioned at the end of the talk that can work well with it.
Yeah, what I learned a bit from Gilead is that chemical modifications are like mutations. Most are detrimental, a few are neutral, and a very few are beneficial. And so the trade-offs are almost always occurring. So I'm not there. I'm not involved in their drug development program. I have no financial support from Gilead. I would be shocked if they weren't looking at multiple methods of development, right? They're trying to directly target the lungs, trying to get this in an oral administration form, because I think if this efficacy works, there will be an exceptional number of people interested in the base chemistry of this and how to modify it, while retaining the capacity to get into cells and be modified. And I think that was the big prodrug advancement, was this phosphoramidate chemistry that allowed for us to have its deliver the parent drug in such high concentrations to the target cells.
Mm-hmm, mm-hmm. And so I'm wondering, if you put it together, where do you see us a year from now? If I know we're all hoping that the vaccines will have an impact, but usually that would take longer.
I guess I'm going to, the scientist in me says everything takes time, and we're working in I tell my lab oftentimes on a daily basis, I say, this is too urgent for us to go fast. We have to stick with trying to understand the basic biology and the modifications of this. But I think that if we can eschew and get away from this sort of let's fire every bullet and throw all the spaghetti against the wall into let's organize around rational teams to look at combinations and try to do this using approaches that we know work and models that we know work, I think we can make very rapid progress. You know, we published that the resistance mutations to Remdesivir cause an increased sensitivity to EIDD 1931. It also targets the polymerase by a different mechanism, but they appear, and vice versa, mutations against EIDD 1931 cause increased sensitivity to Remdesivir. So to me, that's like, yeah, that's interesting. And protease inhibitors as well. Protease inhibitors can fail oftentimes because there's such an absolute selection against them if they're really efficacious, because they're so absolutely required that by themselves you can get rapid loss of their function. But we think that it's possible to, we think those would be ideal. Cell entry inhibitors, so TMPRSS2, protease inhibitors that target the cell. There's so many enzymes, a helicase, hexanuclease, methyltransferases, that are all vulnerable and conserved targets in coronaviruses. And I am discovering that there are people that have worked on these kinds of enzymes their whole lives that have panels of compounds that would be worth testing if we could do this in some kind of a sort of Manhattan project.
Mm-hmm. Yeah, I understand that. Yeah, some of the feline coronaviruses have been treated already with protease inhibitors.
Yeah, they have. And the fascinating disease with a mutation that occurs in a feline coronavirus that converts it to FIPV, feline infectious peritonitis virus. There's a black market in GC376, a protease inhibitor that's been around for a long time now, because it puts those cats into remission. And there are very prominent people in this country and elsewhere who feel that this is the most important mission in the world, to save all their cats. And so there's been a lot of cat testing, anecdotal, independent, but also some studies that support that this protease inhibitor works in cats. And so there's definitely been animal studies, they just haven't been organized.
Mm-hmm.
Mark, can I ask if you're trying to leverage almost kind of the error rates in mutagenesis, yeah, right? But that works if the drug can drive the virus to extinction.
Yeah.
But if it actually requires some interaction and requires host clearing, then aren't you actually promoting its ability to escape the host?
No. No. I'm only saying, sorry, that sounded really absolute, didn't it? So our data says that. So the base for that would be inactivating the hexanuclease, right? And this comes up all the time because you have this 20-fold increase. You have a mutator phenotype, which makes everybody really, really nervous, no matter what we say. So we had to do all of those studies, and what we showed in our lab is passaging that virus for a year in culture. It adapts a bit. It never reverts those mutations, and it's always impaired. In the animal models, Ralph and others had passaged it in immunocompromised and immunocompetent mice of 60 to 90 days, persistent infection, multiple passage experiments, and the virus never gained any fitness advantage and never adapted or became more virulent. So it looks like and so we're interested in this for a couple of reasons. One is it's hard for us to explain from a mutagenesis perspective only how this is so efficacious in single rounds of virus replication, because typically mutagenesis I mean, if I increase the mutation rate 100-fold with the drug, I'm still barely exceeding the XON minus virus, and there's still 30,000 nucleotides. So I think that the evidence is that it's not doing that. So and we think there's other effects on virus replication that are occurring because we're not sure mutagenesis alone can explain it. The other thing is we have evidence that it really profoundly impairs the infectivity of virus particles that come out. So they actually are much less infectious. And so that it makes sense, right? If you're packaging defective genomes or otherwise mutagenized genomes, that virus actually may be less capable of causing disease in the next people it encounters. So there may be beneficial effects we wouldn't expect.
Although to play devil's advocate, right, once you go into humans, it's more complicated than an in vitro system, right? So it could be escaping, antibody responses and those kinds of things.
But remember, mutations are stochastic no matter what, right? And they're also, almost all mutations are detrimental. So I'm deviling back at you, right? So almost all mutations are detrimental or neutral and not beneficial. And if you're going to select a beneficial mutation, it has to have another advantage as well. Right, and theoretically, so we think about this in terms of if we're doing in vitro evolution, that if we put a particular evolutionary selection, a high level of selection, like in vitro, like an antibody or a drug, do we see a difference in the exonuclease minus virus or in the presence of the drug that more rapidly evolves it? And we never see that in our limited systems or over passage, even over a long-term passage with the compound. We don't see an advantage compared to wild type. So, but you're right. We're not doing an experiment with 7 billion people, all of whom have the drug and they're developing immunity. I think the risks are much higher, honestly, for natural selection against those things. That's a much more effective mechanism than anything related to the drug. And if it can happen, the virus, you'll see it. The question is, does it matter?
If I could follow up quickly, so there's been some noise in the news about mutations and strains on the East Coast versus the West Coast. Do you think those are at all medically or clinically significant?
This is a Swiss Army knife. I mean, we're of a virus. We're seeing if you believe it, and I'm putting it on my list of remains to be proven, but some of it's compelling. A thromboembolic disease, a Kawasaki-like disease, people's toes, renal disease. It's exploring human biology, and right now we're a playground. We have to think about this non-anthropomorphically. We have to think about this in terms of, just purely a selection process. And I don't see that the virus is under any evolutionary selection in humans. So as it's exploring our biology, those would have to have a fitness advantage, and increased virulence would have to be some form of advantage. We're doing a natural history experiment, almost Darwinian at this point, right? And until there's some herd immunity and some pressure on the virus from either drugs or some kind of immunity, or there's just something that's even better than this virus, which is a really scary thought. There's not pressure on it. So I think these are more geographic markers right now of diversity than they are virulence markers. But I think that remains to be seen.
Mark, here's almost a philosophical question, but has very practical implications. A lot of people with this infection now, and even if numbers are going down, possible resurgence, but limited supply of remdesivir.
Yeah.
How would you how would you use it?
Okay, so now I'm supposed to do the basic biology and determine how we disperse this drug. Okay.
And who right? Based on who you predict would benefit.
Please stop recording. No. It's really hard. If I could divorce myself from all the things, my first target would be those people who are an at-risk population, who have moderate disease, who are at high risk of progressing, or are hospitalized and high risk of progressing. That would be my first choice here, because I think, or earlier in that process, because you have assuming that the data bears that out, you have the highest risk of having the most benefit for the most people. And then of course, I think also those people who are severely ill, who have a potential for survival, would probably be the first places I would go. And then but we have to be able to keep getting data and information on the range of severity, and thinking about people and populations of people who I can make a list. Those are on chemotherapy and getting infusions. Those are those people who are just come into the hospital but are a high risk group and aren't really even very seriously ill, but you have a probability of actually preventing their transmission, decreasing hospitalizations, decreasing use. there's the social and cultural consequences of it as well. And limitation of supply is sort of the next stage in a pandemic. You know, it's the toilet paper at Walmart. You know, how do you hoard it? How do you use it? Where do you go? Who gets it? And what do we do if we preserve it for one group and one hospital has it in one city and one not in another? I think these are huge social issues.
So then I guess I'll leave you on one last. A lot of folks want to hear about your exonuclease pipeline.
We don't have a pipeline. What we have is we have a tremendous amount of basic biology, and we know ways that we can inhibit it. It's very, very early, but we have early evidence that chemical modification of it can increase sensitivity to these things. Most of those are not things that would ever be used in humans. They would be toxic in human cells. They've been used for other purposes. But I think there's a lot of people we're discovering who have interest in programs and have been working on exonucleases and other viruses and other things for many, many years. And so we're hopeful that that will rapidly accelerate and we can look at that in combinations.
Because of time, I think we have to end. So Mark,
Okay.
Thank you so much. There's so many more questions. There's a tremendously long list that we were not going to get through. But
well, let's see. I'm not doing anything this afternoon, so call me back. No, it's my pleasure. I'm really honored to be here, and thanks for
we
for having
promise
me.
the next time we have you, it's going to be in person.
Okay. Well, that would be fun. I'd love to visit. So thank you very much.
And thank you, Ben, for hosting.
Yeah, and thanks. I really want to thank you again. I know you're so busy, so thanks again for spending the morning with us.
Thank you, everybody.